Glycolysis is the metabolic pathway that converts glucose into pyruvate. After glucose undergo glycolysis, there are three final products produced: 2 pyruvate, 4 ATP, and 2 NADH. So after glycolysis, there are 3 possible fates for pyruvate: anaerobic glycolysis, aerobic oxidation, and anaerobic fermentation. So there are 2 phases in glycolysis: phase 1, preparative phase, and phase 2, ATP generating phase. During this phase, conversion of glucose into glucose 6-phosphate occur and catalyzed by the enzyme hexokinase. The next step is glucose 6-phosphate rearranged into fructose 6-phosphate by an enzyme called glucose phosphate isomerase. A glycolytic enzyme, phosphofructokinase, changes fructose 6-phosphate into fructose 1,6-biphosphate. And then fructose 1,6-biphosphate then split into 2 sugars that are isomers of each other by enzyme aldolase. The enzyme triphosphate isomerase rapidly interconverts molecules dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Next step is phase 2, which is ATP generating phase. Glyceraldehyde 3-phosphate dehydrogenase, GPDH, dehydrogenates and add an inorganic phosphate to glyceraldehyde 3-phosphate, producing 1,3-diphosphoglyceric acid. Phosphoglyceric kinase enzyme transfer a phosphate group from 1,3-diphosphoglyceric acid to ADP to form the energy ATP and 3-phosphoglyceric acid. The enzyme phosphoglyceromutase relocates the P from 3-phosphoglyceric acid from the third carbon to the second carbon to form 2-phosphoglyceric acid. The enzyme enolase removes a molecule of water from 2-phosphoglyceric acid to form phosphoenol pyruvic acid. By the end of the glycolysis reaction, the enzyme pyruvate kinase transfers a P from PEP to ADP to form pyruvic acid. So the overall of the reaction of glycolysis is where glucose added with NAD+ and molecule of ADP, molecule of inorganic phosphate produce pyruvate molecules, NADH, ATP, molecules of water, and hydrogen. Moving on to the fate of NADH and pyruvate. NADH and pyruvate are energy of 2 possible fates: aerobic oxidation and anaerobic conditions. In aerobic oxidation, oxygen molecule is available. Pyruvate molecule loses a carboxylic group in the form of carbon dioxide. After pyruvate molecule loses carbon dioxide, the remaining 2 carbons then transferred to the enzyme CoA to produce acetyl-CoA. In oxidative phosphorylation, NADH reoxidizes in the electron transport pathway and produce ATP. In anaerobic conditions, oxygen is absent. NADH is reoxidized to NAD+, which then provide additional NAD+, for more glycolysis. In anaerobic glycolysis, or also known as lactic acid fermentation, whereby pyruvate converted to lactate and the NADH is reoxidized to NAD+, which then provide additional NAD+ for more glycolysis. Fermentation to lactate in vigorously contracting muscle, erythrocytes, some other cells, and in some microorganisms. In anaerobic alcoholic fermentation, the pyruvate loses carbon dioxide and form acetaldehyde. And then from acetaldehyde, then converted to ethanol, whereby the NADH oxidized to NAD+.
This video will explain about the Kreb cycle, or known as the citric acid cycle. So in this cycle, the reaction will be start with the conversion of pyruvate into the acetyl-CoA. So in this reaction, so it involves the removal of the carbon dioxide, and during the removal of carbon dioxide, the energy is given off, and the NAD+ will be formed or converted into the higher energy, which is in the NADH form. So the coenzyme A will attach to the remaining 2 carbon unit, which is the acetyl, to form the acetyl-CoA. And the acetyl-CoA will be then react with the oxaloacetate, which will form citric acid. That will be the beginning of the Kreb cycle. The citric acid will be then transformed into isocitric acid when reacted with aconitase. So between isocitric acid and alpha ketoglutarate, the NAD+ will be converted into NADH. Meanwhile, there will be a release of carbon dioxide while the reaction proceeds. The next reaction will be between alpha ketoglutarate and also the succinyl-CoA. So in this reaction, NAD+ will be converted again into NADH, and also it involves the removal of carbon dioxide. Next is the conversion of succinyl-CoA into the succinic acid. This reaction involves the formation of GDP into GTP by the transfer of energy. Meanwhile, it releases CoA, which results in the formation of succinic acid, which is also known as succinate. The next reaction is about the conversion of succinic acid into the fumaric acid. So, between this reaction, the molecular shift releases not enough energy to make the ATP or NADH. Thus, this energy is captured by a new carrier, which is the flavin adenine dinucleotide, which is called the FAD. So, during this conversion, the FAD will be reduced by the addition of 2 hydrogen atoms to become the FADH2. Then, during the formation of the malic acid, there will be addition of water and the presence of fumarase, which result in the conversion of fumaric acid into the malic acid. In the last reaction of this cycle, there will be a formation of the oxaloacetic acid. So during this reaction, the malic acid will be formed into oxaloacetic acid with the removal of NAD+, which then will be transformed into NADH. Then the result will produce the oxaloacetic acid, which will react with acetyl-CoA and then start the cycle again, which forms the citric acid.
In the electron transport chain, electrons are being passed from one molecule to another, and the energy released in these electron transfers is going to be used to form an electrochemical gradient. The electrons will then provide energy for the pumping of protons into the intermembrane space from the mitochondrial matrix. At the end of the process, two water molecules will be produced. Shown in this diagram is the membrane of the mitochondria. This is where electron transport chain occurs. It also involves intermembrane space and the mitochondrial matrix. The matrix contains protons in the form of hydrogen ions. The membrane of the mitochondria consists of complex 1, 2, 3, 4, coenzyme Q, and cytochrome C. The NADH produced in the previous process is going to go through redox reaction and produce NAD+. The electrons produced in the redox reaction will then move to complex 1 and make complex 1 supercharged. Complex 1 will then have enough energy to pump protons into the intermembrane space from the mitochondrial matrix. It will then produce a gradient. The electrons in complex 1 will then move to coenzyme Q and stay there for further action. The FADH2 produced in the previous process will be reduced to FAD. The electrons produced in this redox reaction will then be moved to complex 2. But complex 2 will not be supercharged like complex 1. So, the electrons will move to coenzyme Q and stay there for further action. Next, the two electrons will move to complex 3 and makes the complex 3 supercharged, so it will have enough energy to pump hydrogens into the intermembrane space and produce a gradient. Next, the two electrons will move from complex 3 to cytochrome C and next to complex 4. The two electrons will make the complex 4 supercharged and thus it will pump the protons into the intermembrane space and produce a gradient. The two electrons will then move out from complex 4 and react with oxygen. It will then produce two water molecules. And that's all for electron transport chain. Thank you.